afternoon, good day, everybody. Today we're talking about cell division and differentiation. So why do we need to go through all of that? And how does it all work? What's it do for us? What's it do for just life in general? So, as we talked about on your vocab words, we looked at some pictures of different onion root tip cells the other day. What is that process that starts with one cell and ends with two identical cells? One of them in eukaryotes is mitosis. And mitosis turns one cell into two identical daughter cells. So the important part, they're identical. They should have all the same parts, all the same chromosomes, all the same DNA. And mitosis is just one short part of the cell cycle in general. There's lots of different parts. So we have G1, S, and G2, and G0. And last couple of weeks we were talking about DNA, and that happens during that S phase. So if we start at the beginning, if you start to think of the cell cycle as two big parts, you have the first big part is interphase, and then the second part is mitosis. Now within interphase, there's some growing that happens, and then there's also preparation for copying. So in G1, the cell cycle, things are getting bigger, you're making copies of your organelles, you're doing your normal functions of that cell. That's where most of the cell's life is spent, in that G1 phase. Then in S phase, we copy that DNA because our new cells are each also going to have a copy of the DNA. Then in G2, we check the DNA for errors, finish any growth, and some cells go into G0. We'll talk about that in a little bit. So things like neurons, that sort of thing, they don't make copies of themselves. They just get to where they need to be and they stay there. So one thing you should check is what we just talked about kind of has to do with this, to illustrate and describe events of each phase of the cell cycle. So you can pretty much take a picture of this, draw that in, and then go ahead and continue working on your targets, get that finished. So our next thing, check your notes, make sure you know what mitosis is, what does mitosis create, how much of a cell's life cycle is spent in mitosis. You might have to do a little research there. I mean, like percentage. Is it a big part, is it a small part? And then what is the cell doing when it's not dividing? So that goes right back to the picture we just looked at. So you should know that mitosis has three main functions. The first one, allow the organism to grow. So one thing we talk about is that cells are all small. So if you need to have a big organism, that cell just doesn't become giant. And that has to do with the ability to absorb nutrients and things through diffusion and osmosis. To get bigger, you have to just get more cells instead of just growing one giant cell. Mitosis can be used to repair tissue. So if there's a tissue type or cells were broken or damaged, they need to copy good ones and mend it. So different tissues can fix themselves. And then single-celled eukaryotes, we've talked a lot about yeast this year, that'd be an example. They make copies of themselves. So an amoeba will just copy itself, it'll clone itself, and that happens through the process of mitosis. So let's go back right quick. And if you look at your targets, we have a target about that. Define mitosis and list three examples of cells that undergo mitotic division. I gave you the example of yeast. All of your cells will do that. And then come up with another one. Then list two purposes of mitotic division for multicellular organisms. So multicellular, you, me, a plant, that sort of thing. Not the yeast. The yeast is single cellular. Alright, so going forward, how does it all happen? Now, this gets a little bit more advanced than what you're going to be responsible, but we really feel that in order to really understand this process, you have to see all of the steps. So, again, interphase is actually not part of mitosis. Prophase is going to be our first step. So in interphase, notice we have our nucleus, and the DNA is all loose in chromatin, right? In prophase, notice we don't have any nucleus anymore. It's disappeared. That nuclear envelope has disappeared. And now, instead of being strands of DNA, it's organized into these chromosome shapes. And that's part of organizing. Think of it like you had all the instructions of that cookbook. Right here, all the pages are scattered on the ground in the kitchen, and you can read them all really easy to express things, but it's not organized. And you want to bring them different places. So if you want to bring them different places, you organize them into different volumes. And those different volumes are your chromosomes. Nice and organized, easy to move around. In metaphase, now we need to match them with their pairs. So right here we have one chromatin, and here we have one chromatid. Right? Together they make a chromosome. So they're organized in the middle, and they're going to get pulled apart. So they get pulled apart to either side in anaphase. And now the nuclear envelope starts reappearing around each set of chromosomes, and eventually through cytokinesis, when the actual cytoplasm pulls apart and the different organelles pull apart, we get two new cells. And important, these two cells are the same as they were at the beginning. Right? So you have prophase, metaphase, anaphase. When they're breaking apart, we have telophase and cytokinesis. And then here we have our two new cells, and we'll go through over and over and over. So in mitosis versus binary fission. Mitosis, what we said, eukaryotes will do that, things with nucleus, because a eukaryote has a nucleus, and that mitosis is that splitting of the nucleus, splitting of the chromosomes from side to side. In binary fission, that's just for prokaryotes. So in prokaryotes, we go through a little different process. They don't have chromosomal DNA. Prokaryotes don't have the nuclear envelope. So we have this plasmid here that's a circular copy of the DNA that we find in prokaryotes, and it copies itself really fast. It goes through replication like we practice in class, where you just match up those A's, T's, C's, and G's. It's super duper fast. And sometimes there's errors here, more often than eukaryotes, so they don't check for those errors. And then we split the cell. We don't have to go organize all the chromosomes because we just have this one plasmid, this one copy of DNA. So in mitosis, we have organized chromosomes. In binary fission, we have the unorganized plasmid. That's just a simple copy. It happens fast. But again, the idea, we're making copies, right? So go ahead, check your notes. So far, you should have what are the three purposes of mitosis, what are the stages of mitosis, and what do bacteria use instead of mitosis. As far as what are the stages go, that's good to know, but it's not going to be something where you have to identify. Just make sure that you know we're organizing that DNA into chromosomes so it can be pulled apart to different sides. So if we look at our targets now, let's see, listed purposes, yes. Compare and illustrate DNA is found within a prokaryote and eukaryote cell and how the DNA is organized during that cell division. So here's where you talk about those chromosomes versus that plasmid and talk about organizing those chromosomes, pulling apart, breaking down the nuclear envelope versus just making a quick copy of that plasmid, right? That's what that target's about. Then right here it says explain how copies of genetic material are transferred from parent cell to daughter cell through the process of mitosis. That would be a good place to go back and look at this picture right here. You could draw, I really like this, where you have metaphase, have a picture of metaphase, picture of anaphase, and then two final pictures, something like that. That'd be a really good picture to draw, or some version of that. All right, now we get into cell differentiation. This is a little bit more complex, but we're going to make it really simple for you to understand. We're not going to know about all the different checkpoints and what decides what. That's something that a lot of scientists are still working on right now. But the main idea is each cell type is a little bit different. We're going to express genes differently, and that's going to be through cell differentiation. Different cells will decide to become different based on where they all started. So remember, each cell has a copy of all the DNA. So any cell has all the instruction for how to make all these different types of cells, all these different types of tissues. But that's decided early on in the development of the organism. So differentiation is this idea that one cell can become many different types of cells. How does cell differentiation differ from mitosis? Well, mitosis is just making copies of the cell. Differentiation is becoming specialized. So, let's go take a look here. Here I have my single cell. This is undifferentiated. All right, it's not become specialized yet. It can become any sort of cell. Different environmental cues, different cues by hormones, different cues within the DNA themselves will tell the cell what exactly to do. And remember, the DNA has genes, and there's lots of different genes. They tell you how to make proteins, how to make a new cell. So this one cell could become something like a neuron, and this one cell could be something like an epithelial cell, a skin cell. Very different cells, but originally they were the same, and they all had the same DNA. Right? So we want, in differentiation, just think different. We're becoming specialized. So, check. Do you know what cell differentiation is, what it does, and what genes have to do with that differentiation? Remember, genes are what make things get expressed, our phenotype, and that's how they become different. So what part of the DNA gets expressed changes what type of cell you get. So let's take a look at our notes again, our little targets. Let's see, go through here, and we have explain the relationship among, best goes with these right here is going to be explain why cells need to differentiate. Give an example of cells that can differentiate into multiple cell types. So we didn't really talk about what types of cells can differentiate. So you've probably heard of stem cells, those early cells in the development of an organism that can become lots of different things. So we have a little picture here. A zygote, you should know, is a fertilized cell, so a fertilized egg that becomes a zygote, and it's going to copy itself through mitosis. And these are all exact same generic cells. And they're still exact and the same. Nothing's really happened. So we have a lot of cells, they start making a ball, they're still all the same, and then we have this process of gastrulation where we start folding in on this cell, and we're going to get different germ layers. And at this point, they're all still not quite different. They're all generic. Right at this point, we're starting to become specialized. So as far as types of cells that can become different things, stem cells, or we have the zygote, or we have these germline cells. And you can see these early cells, you have different layers. You have the ectoderm, which is this outside layer, the mesoderm, which is this pink middle layer, and then you have the endoderm, which is the inside. And these germ layers early on in development can become different tissue types, all listed below here. So you could use any of these as an example. You could say the mesoderm could become red blood cells or bone tissues. And again, this is if you're talking about people when they are in uterus, all right? So as they're developing, early on, first few weeks, first month, okay? That's what we're talking about. Now, take a look at your notes. From fertilization, what are the steps of a gastrula? You're not going to need to know this, but it can help you explain that idea of differentiation and what sort of cells can do that, or some specific cell types that come from each germ layer, same sort of thing. It'll help you understand and explain that idea of differentiation, how some cells become different cells because of the DNA. And that's it. Go ahead and make sure that you can write a nice summary, come back, write some questions, chunk it, try and come up with some different test questions, star vocab, and work on addressing those targets. Thank you, everybody. You have a good day.